Hello guys. Welcome. Today we are going to continue where we ended yesterday. The usual structure is prepared for you. Introduction activities, and then the core, some intro to the um, or some general intro into the genetics when we are done with the introduction, and um, then focusing on Mendelian genetics and Mendel's law. So this is an agenda for today, basically, and I would like to ask you if you have any questions before we start regarding whatsoever. No questions? Okay, so let's start then. Let's start with the introduction. Um, Here we have a first. Sorry, my bad. Here we have a first activity, which is matching activity. So your task will be for upcoming 2 minutes, or let's have 3 minutes. Let's have more time because there are five definitions. So let's have 3 minutes. So individually try to find the respective definition to the term from genetics. We discussed all of them already together day before yesterday, and also yesterday. So this is your task for upcoming 3 minutes. uh, you will try to find the respective pair to the or respective term to the definitions on the slide. So let's start right now. Sorry guys, there were some internet connection issues on my side, so I apologize. I was away for a few seconds. Okay, nevertheless, are you done? Can we start with the first phenotype term? Anyone?
Phenotype, I think the set of observed identifiable characteristics of an individual resulting from the interaction of its genotype with the environment.
Perfect. Great. Now, can you link it, please? Great. Thank you. What about next one? Genotype. Anyone? Have you found something?
I think genotype is refers to the specific genetic makeup of individual with respect to a particular gene or set of genes.
Great, Choco. Please, can you do the same and link it? Mm-hmm. Thank you very much. Okay, genome. What about genome?
I think genome is refers to the complete set of genetic materials or DNA present in an organism's cell, encompassing all of its genes and non-coding sequences.
Perfect. Perfect. Thank you, Misato. Can you do the same and link it?
Yes,
Thank you very much. What about allele?
I think
Whoever wants.
uh, I think allele is each of two or more alternative forms of a gene that arise by mutation and are found at the same place on chromosome.
Very well, Satsuki. Great. Can you link it, please?
Yes.
And the last one, gene.
Gene is a unit of heredity which is transferred from a parent to offspring and is held to determine some characteristic of the offspring.
Thank you, Shoko. Great. Please link it. Great. Okay, guys, so all of those terms we already discussed in the previous sessions, and now it was just a short recapitulation. Um, if some of you is still not familiar with the definitions, so those are possible definitions, and if you are still struggling with them, do a quick screenshot now. So basically, you will know the respective definitions of those terms because those definitions of those terms are very very important for the upcoming days or upcoming sessions, at least for few which will be more focused on genetics. Okay, so please be familiar with those things in order we do not have to spend a lot of time in the future with recapitulating what does the phenotype or genotype or allele or genes or genome as well are, because it's very essential for the genetics as a topic. So hopefully all of you uh, have noted down what those terms refers to, or what do they mean, and that brings us to the quiz. Yuki, are you here?
Yes.
Perfect. Please, can you read?
What is the primary purpose of meiosis in sexually reproducing organism? A. To generate genetically identical offspring. B. To increase the number of chromosomes in the offspring. C. To produce the
Gametes.
gametes with,
Gametes.
with
Great.
gametes with half the number of chromosomes. D. To create somatic cells for growth and development.
Thank you very much. Yuki, have let's have a few more seconds. Three more people. One. Okay, so here are the results. Yuki, what have you chosen and why?
I choose C.
Hmm.
So, mm, because I thought A, B, D is not correct. So A is so uh, so genetically identical offspring is not correct.
Perfect, exactly. Because genetically identical cells are the outcome of what? Not meiosis, but genetically identical daughter cells are outcome of what? Anyone?
Mitosis.
Perfect, mitosis. So this would be correct if we talk uh, about mitosis. Great. What about B?
Uh, to increase the number of chromosomes is not correct.
Perfect, great. We're not increasing the number of chromosomes because this would be already a numerical mutation in the cells, so there will be definitely some um, physical or basically some health conditions which would be definitely different comparing to healthy patient or a healthy individual, let's say. Uh-huh. What about D?
D. So for growth and development.
Growth and development is very general. We can say that meiosis could be or like meiotic 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 cells or the gametes after meiosis too. They basically play a crucial role in fertilization an egg and then a zygote, and basically then the zygote is growing and developing and creating a human being. So this part is not like 100% incorrect. What's the problematic is another part. Like this is very hypothetically possible or related to meiosis, let's say. The other part, what is definitely not an meiosis purpose in the D.
Meiosis purpose is produce a sex cell.
Perfect. Sex cells or gametes. Okay, so definitely not somatic cells. Okay, meiosis do not ever produce somatic cells. We use an as an example if we have, for example, dog or cat or any other pet which is neutered and doesn't have any more reproductive organs. No meiosis is going on inside its body. Okay, so it is highly specifically related only to gametes or sex cells. Okay, meiosis. Very well. Thank you, Yuki. Let's go on. Misato, can you read next one?
Yes, which of the following statement best describe the outcome of mitosis? A. Formation of haploid daughter cell with half the number of chromosomes. B. Production of gamete, sperm and egg cell for sexual reproduction. C. Generating of genetically identical daughter cells with the same number of chromosomes. D. Creation of somatic cells with unique genetic combinations.
Thank you very much, Misato. So let's have a few more seconds to read it one more time, everyone. And then we will do a quick poll. Okay. Still a few more people. Three more. Two. Just one. Okay. So here are the results. So all of the options were chosen, so let's discuss them, which one is correct and which are not. Misato, please, what have you chosen and why?
I chose D because my mitosis create somatic cells and so so I and create unique genetic combination. I think is correct.
Okay, so you went for D. Let's propose that D is correct. Um, what's wrong with A, guys? What do you think A is wrong? Formation of haploid daughter cells with half the number of chromosome. If we are talking about mitosis, what doesn't fit, in your opinion?
Half the number of chromosomes is incorrect.
Perfect, exactly. Because if we remember, and I will show the picture, the outcome of mitosis and meiosis are two daughter identical cells in mitosis. However, in meiosis, we have four haploid
not identical, but different cells. Perfect. Great. So if we see our question one more time and we see that which of the following statements best describes the outcome of mitosis, A statement definitely is not the right one. Formation of haploid daughter cells with half number of chromosomes would be correct if we are discussing the meiosis. So some of you or one person chose A. Hopefully guys, now it's clear why A can't be correct. Okay, because if we are discussing mitosis, definitely there is not an haploid daughter cell as an outcome. There is classic diploid daughter cell as an outcome with the normal amount of or the normal number of chromosomes, what is 46. Okay, so this can be correct. What about B?
B. Describe meiosis.
Great, great. Production of gametes or sperm and egg cells for sexual reproduction. So definitely gametes are highly related only to the process of meiosis. So nothing to do with mitosis. Great. C or D then. Now, who thinks the C? Who thinks D? So Misato went for D, someone else went for C. So guys, what are your arguments? Why you think D or C is correct? Anyone? If we break down the C option, generation of genetically identical daughter cells with the same number of chromosomes. So far it seems good. So let's discuss D. Creation of somatic cells with unique genetic combinations. There is something not correct, something what is wrongly stated regarding mitosis. Do you know which part of this statement is wrong?
with unique genetic combinations.
Perfect. Unique genetic combinations. Because outcome of mitosis are two identical cells, there are no differences. So they are also, however, in meiosis we have four haploid and different cells. In meiosis, the outcomes are the unique combinations of cell, and very probably the combinations in the genetically was the first time also in human species history. So that's why all of us are a little bit different or even more different. That's why we are not just copies to our parents or to our siblings, because crossing over takes place during when, guys? Crossing over, when does it takes place?
Prophase 1.
Great, during prophase 1. Prophase and crossing over takes place. So that's the reason why the offsprings are always unique, and there is practically zero chance that your your genotype or genome inside our bodies are similar to anyone else on this planet Earth, which will have the same genome as we have, basically. So this is the reason. So, however, in mitosis, this is not happening. So that's why in my mitosis, um, there are identical daughter cells as a result of its process. So if we get back, Miss, I don't know, is it clear why D is not correct and C is correct?
Yes.
Perfect. Great then. Okay, guys, so let's move then further with another one. Now, please, can you read?
Which of the following statements best describes interphase in the cell cycle? A. It is the phase where cell division takes place, resulting in two daughter cells. B. It is a brief resting phase between cell divisions. C. It is the phase when the cell replicates its DNA in preparation for division. D. It is the phase when the cell shrinks in size and reduces its metabolic activity.
Thank you now. So let's have a few more seconds. Three more people. One. Perfect. So, guys, here are the results. Let's discuss them. Now, what have you chosen and why?
I'm not sure that my answer is correct, and I was thinking about B and C was correct, and I chose B.
Okay, so you were struggling with B or D. Oh, with B or C. Okay. No one went for D. So what's wrong with D? Let's start with D. Why D can't be correct, guys? If we are discussing cell cycle interface. It is the phase when the cell shrinks in size and reduces its metabolic activity. So if we get back to our cell cycle diagram, we remember that we have basically three phases during an interface, G1, S, and G2. G1 is well known, or the main processes which are undergoing during this phase is basically growth of the cell and growth of the organelles and also the essential substances important for cell division, for upcoming cell division. So the cell definitely is not shrinking. On the contrary, the cell is uh, growing. Okay, and wait a second, let's get back. So it was this. Yes, it is the phase when the cell shrinks inside and reduces its metabolic activity. So also on the contrary, it doesn't reduces the metabolic activity. The metabolic activity inside the cell is normal or even higher than usual, comparing maybe to G0 phase. Okay, so the cell definitely is not shrinking and is also doesn't reduce metabolic activity. Okay, uh, you were struggling with B and C. There was one option for A. One person chose A. So let's discuss A. Is it clear? Is it right or is it wrong? What do you think, guys? It is the phase when where cell division takes place, resulting in two daughter cells.
I think A is not correct because A A statement describe about M phase.
Perfect. Exactly. So it is the phase where cell division takes place, resulting in two daughter cells. Definitely is not interface because the phase when the cell division takes place is M phase. Okay, M phase. M phase uh, contain mitosis, and even some authors divide mitosis into another phase, which is cytokinesis. Okay, during M phase, both of those processes are taking place. So hopefully now it's clear why A option was not correct in our question, guys. Okay, now let's discuss the other option. Interphase as a brief and resting phase between two cell divisions. As we see, the comparison of a time between interphase and M phase is pretty huge in advance for interphase. Okay, so most of the time during cell cycle, the cell is in interphase, so it's preparing, synthesizing new DNA, growing and duplicating also the organelles and the substances, proteins, enzymes, everything what is important and essential for upcoming cell division. So if we get back to our question, now this one, sorry, I apologize. It's definitely not a brief resting phase between cell divisions because there is a lot of going on, and also it's not brief even though time is relative, but uh, comparing to M phase, interphase takes much more time to be finished comparing to M phase. So brief and resting phase is not right statement. However, interphase is the phase when the cells replicates in DNA in preparation for cell division. Okay, so when does the DNA replication happens during cell cycle, guys? Do you remember?
S phase.
Perfect. S phase. During S phase. So before S phase, we have a G1 phase, where we also have the checkpoint, first checkpoint, and after S phase we have another checkpoint during G2 phase to check whether everything is properly properly synthesized and whether the cell is basically in a physiological condition and can continue in mitotic or cell division. Perfect. Okay, so that was this one. Let's continue. Aoi, please, are you here?
Yes.
Perfect. Can you read, please?
What is the primary purpose of translation in the process of gene expression? A. To du- duplicate the DNA code during cell division. B. To convert RNA molecules into DNA for long-term storage. C. To synthesize proteins using the genetic information encoded in mRNA. D. To repair damaged DNA within the cell.
Thank you very much, Aoi. Let's have a few more seconds. Few more people. One. Okay. So here are the results. Howie, please, what have you chosen and why?
I chose she.
C. Okay. To synthesize proteins using the genetic information encoded in mRNA. Okay, so let's propose this is right. So what's wrong with B? To convert RNA molecules into DNA for long-term storage. How do we call this process? Anyone? Do you remember? We mentioned once or twice already reverse transcription. Reverse transcription is a way how the retroviruses or HIV viruses infect the host cells, basically. And it's via reverse transcriptions. This is the process. They convert RNA molecules into DNA for long-term storage. So this is not translation. Our translation we are seeking, this is a reverse transcription. To duplicate DNA code during cell division, how do we call or what is the name of this process?
Replication.
Perfect. Replication, not translation. Replication is when we basically firstly need to unwind the DNA molecule via helicase enzyme, then we synthesize this new DNA strands via DNA polymerase, and on leading strand and on a lagging strand we need one more enzyme we call ligases, which will replace Okazaki fragments in order to have two complete DNA doubles, two complete DNA double-stranded molecules. Okay, so this is DNA replication, which takes place in S phase, as we already know, as we discussed in a previous question. So no, this is not our question. This is our question. To repair damaged DNA within the cell, guys, DNA repair mechanisms are taking place during the checkpoints we have in G1 or G2. If you remember, if there is some damage, DNA damage detected, the cell either trigger apoptosis or the program cell death or stops the cell cycle. And if it stops the cell cycle, the DNA repair mechanisms are trying to fix the issue or the wrongly synthesized DNA, and they try to fix it. If they will fix it, then the cell basically can continue and undergo the cell cycle and went towards the cell division. However, if not, it can also just remain in the position, so there will be no cell cycle going on. So basically, the cell will itself switch to G0 or will basically suicide itself via apoptosis, what is also absolutely physiological process in the organisms. Okay, so DNA repair mechanisms are taking place during at the end of G1 and also during G2 phases. Okay, here we are. So great, guys, and let's do two more. Satsuki, please, can you read?
What is the primary purpose of reverse transcription in molecular biology? A. To duplicate the DNA code during cell division. B. To convert RNA molecules into DNA for long-term storage. C. To synthesize proteins using the genetic information encoded in mRNA. D. To repair damaged DNA within the cell.
Thank you, Satsuki. Okay, let's have a few more seconds. Two more people. One. Okay, here are the results, so let's discuss them. Satsuki, what have you chosen and why?
I chose B because last question I know reverse transcription is to convert RNA into DNA.
Mm-hmm. Great. Uh-huh. So, to convert RNA molecules into DNA. Okay, so what is then A, if not reverse transcription? Or to
A,
duplicate? Go on.
A is replication.
Yes, not during the cell division, okay, is not happening during cell division, but definitely duplication of DNA is replication. So A statement can't be correct. To synthesize proteins using the genetic information encoded in mRNA, how is this process called?
Translation.
Perfect, translation. Where does it take place? Take translation, do you remember? Anyone?
Ribosome.
Perfect. Ribosomes outside from the nucleus. Here we see it. Here we have ribosomes, our strand of mRNA, and tRNAs with their anticodons are just landing on our RNA strand in order to then synthesize polypeptide chain and our proteins we need. Perfect. Uh-huh. Okay, so here we are, I guess. This is it. Yes, no. This one. To convert, to repair damaged DNA within the cell. So definitely, reverse transcription doesn't do that. It converts RNA molecules into DNA. Let me very shortly explain how does that viruses are doing. So we have an RNA strand. RNA strand uses reverse transcriptase, reverse transcriptase enzyme in order to synthesize DNA strand. This single strand, we have only single-stranded DNA, is able then to get into host cell. Okay, so I will just draw host cell. Here we have a nucleus, and this DNA, single-stranded DNA, is able to get into the host cell and basically modify the genome of the nucleus and of the organism. So, for example, HIV viruses infect the host cells, and the manifestation, or basically they're changing DNA, and those changes in DNA are decreasing activity of immune system. That's why HIV patients are more probably or likely and much more often infected via common infections such as um, other viruses, and they could very easily die of pneumonia or or urine infections, which are very common, and um, or let's say healthy patients have no problem to resist those common infections. However, HIV people are more likely to be infected and affected via those common infections. That's only because the HIV virus have an reverse transcriptase enzyme which then synthesize DNA, and that single-stranded DNA gets to the host cell nucleus and modifies and change the genome, basically the genetic material inside the human bodies. Okay, so that is the process. You don't have to be familiar with, we don't have to remember reverse transcriptase and the things, but please remember that HIV virus is an example of retroviruses, and retroviruses uses or are using reverse transcription. Okay, reverse transcription. Basically, transcription is the transcription just in the opposite direction. Okay, in the usual transcription, as we learned here, we have our DNA molecule, and then RNA polymerase synthesize mRNA. Okay, so from DNA we have RNA. In reverse transcription, we synthesize DNA based on RNA template. Okay, so it's just in the opposite direction. Okay, understandable so far, guys? Perfect. Okay, let's have then one more, and that's it. Yuiko, please, are you here?
Yes.
Please, can you read?
Okay. Which of the following statements best describes the role of the cell nucleus in eukaryotic cells? A. It is the site of protein synthesis and RNA production. B. It regulates the entry and exit of materials into and out of the cell. C. It stores genetic information and controls cell activities. D. It is responsible for breaking down cellular waste materials.
Thank you very much, Yuiko. Okay, so here are the results. Yuiko, what have you chosen and why?
I choose C.
Mm-hmm. Perfect.
Uh,
So it stores genetic information and controls cell activities. Okay. What organelle is responsible for B option? It regulates the entry and exit of materials into and out of the cell.
Membrane.
Perfect. Very well. Cell membrane would be the option here. Cell membrane. Uh, what about A? At least to protein synthesis. What do you think?
Ribosome.
Perfect. Ribosomes. And the last one?
I don't remember.
Okay, anyone? Anyone?
Rhizosome.
Lysosomes. Perfect. Lysosomes would be responsible for that. Lysosomes. Perfect. Okay, guys. So it's 18:01 your time, right? Perfect. Would you like to have a break? Okay, so here are the results. All of you want a break. Great. Okay, guys, so my time. Okay, so it's 18:02. So let's meet at 18:12 here back. Okay? See you.
Okay, guys. So time's up. So let's start with the um, genetics where we ended yesterday. Sorry for the typo. I tried to correct myself uh, in the genetics headline. Nevertheless, I would like to maybe just very briefly introduce you to the genetics we haven't covered yesterday, and then we will go further to the group work you yesterday started with. So I prepared something, so let me just present it, and there will be some important information you should note down because some of those information also can occur in the admission questions. So, genetics, or brief intro to genetics. People have long observed the inheritance of certain traits and use it for the breeding of plants and animals. However, the rules governing inheritance were not revealed until Johann Gregor Mendel did so in 1865 when he presented his findings in Brno. So for those who are interested in joining and applying for studies in Masaryk Foundations in Brno, this can be interesting because there is another university named after Johann Gregor Mendel, Mendel's University. It was not enough for the scientific community back then, and his discoveries received recognition only in 1900. Long after Mendel's death, globally his perspective was acknowledged in 1925. Many opponents cited inherited traits that partially or did not conform to Mendel's rules. These deviations were later explained by a detailed description of chromosomes, meiosis, and especially crossing over we already know. Other variations were clarified by polygenic or multifactorial inheritance. The assumption that DNA is the chemical basis of genetic information dates back to 1927. Convincing evidence was provided in 1952 during a race to describe the structure of DNA and unveil the secrets of genetics. British scientist Rosalind Franklin came closest to victory. Let me show you the picture. In 1953, she captured famous image 51. This is it. Let me show you. This is the picture and this is the scientist. So, which demonstrated that DNA has a double helix structure. She shared her discovery with her colleague Francis Crick and shortly after passed away. It's very important or also very sad story because she played a crucial role in understanding the DNA molecule, but she was not awarded with Nobel Prize. However, in shortly after, the Nobel Prize discovery was later awarded to Watson and Crick. Uh, Guys, please remember there is sometimes in the admission questions that who was awarded with Nobel Prize uh, for discovery of DNA structure. Rosalind Franklin was not awarded, but Watson and Crick, Mr. Watson and Mr. Crick were. Okay? Rosalind Franklin unfortunately did not receive Nobel Prize because Nobel Prize is not awarded after a death. Okay? So, only Watson and Crick received or were awarded with Nobel Prize. And these kind of questions sometimes occur even in the admission questions. So please note this down.
In the following years, scientists worldwide focused on identifying genes as specific segments of DNA in experimental organisms and humans. The endeavor to read the human destiny was partially fulfilled in 2000 when the results of the Human Genome Project were published. Complete results were available from 2003. Since then, the use of DNA analysis has significantly expanded for determining predisposition to certain diseases for forensic purposes, paternity testing, assessing individual drug efficacy, and for detailed studies of evolution. There have been successful attempts at treating genetic disorders in the 2000s. However, the knowledge of Mendel and classical genetics laws remain essential and relevant even for today. And that brings us to our topic, Mendelian genetics. We started with this topic already yesterday with the group work, and now it's time to describe and to discuss the first Mendel's law. First Mendel's law, let me show you the slide. And I would like to ask the group from yesterday, what are your findings, guys?
Mendel's first law is that when a pure line parent with a dominant trait is crossed with a pure line parent with a recessive trait, the offspring will have only the dominant traits.
Mm-hmm.
That's all.
Perfect. That's very correct. Thank you, Shoko. So let me maybe just provide you with a little bit more information. Um, During Mendel's time, the practice of crossbreeding was very common, and cultivators used pure lines, which were basically homozygous for the observed traits. Okay, pure lines, homozygous. Okay, we have here dominant homozygous and recessive homozygous. For example, if they grew 100 pea plants, pea, do you know what is pea? Um, I will try. Let me give how to say. hmm 1 second. Pea. It's
Endo,
endo. Endo. It's pea. You know. Perfect. So if the cultivators grew 100 pea plants, and 10 of them were yellow, they simply discarded those and continued the process. In the next following generation, there were only 5 yellow plants. And so, until they achieved 100% of the desired green pea plants, this process continued. This process did not require any knowledge of genetics. That was already known before the genetics was as an individual scientific field. However, Mendel approached crossbreeding with a scientific perspective. He worked with 3 generations of pea plants. When the first parental, we will draw P here, first parental generation always came from 2 different pure lines. What does it mean? Basically, they are homozygous. The first one, dominant, in our case yellow P, is dominant homozygous, is from different pure line than the green homozygous with recessive alleles. Okay, so it's from different pure line, but they are homozygous. They share same trait uh, in the generations. That's the pure line. He crossed these pure lines to create the first filial generation and then propagated them to produce the second filial generation. Okay, so here we have first filial generation, and in the upcoming, here we have second filial generation. We will get there very, very shortly. Uh, Mendel noticed several recurring rules. In the first filial generation, here, all plants were same or uniform. That's why the first Mendel's law is called the law of uniformity of F1 generation hybrids. In the second filial generation, the plants showed variations from their parents and each other So we call it segregation. So there are differences between offsprings and differences between offsprings and parents. Additionally, he observed that some traits were either fully expressed, we will now get there, fully expressed, or not at all, while others showed an intermediate blending, in this case, incomplete dominance. So, on left side, we have a complete dominance between alleles. On the left side, we have incomplete relation between alleles, incomplete. We are now going to discuss them. The laws are stated in their verbal form, and we will demonstrate each of them with images for complete dominance and incomplete dominance for simplicity. For simplicity, we will focus only on one trait, okay? So, let's get back to our first law. Law of uniformity is when two homozygous individuals are crossed and the offspring are genotypically uniform. What does it mean? Genotype of one parent is AA, big in bigger case letters, and the other one is recessive. We have lowercase. On this side as well, we have to remind us, I will use a different color. Let me use different color. We have a complete relation between alleles. On the left side, incomplete. What is the outcome? Genotypically, the offspring, if we cross those gametes, the father and the mother basically, A, the big A will be in all of the offspring, and also the small a will be in all of the offspring. What does it mean? The gametes from father and mother were combined, and all of the offsprings of two homozygous will be uniform, but they will be heterozygous. In complete dominance, Mendel's discovered that the dominant allele, in our case big A, will mask the presence of recessive allele. That's why all of the offsprings are yellow, okay? Because the complete dominance um, hides or masks the the allele for green color of the piece. However, let's now move to the right side. In incomplete dominance, when two homozygous parents are crossed, both alleles are blended, or let's say the trait is blended, and they are, let's say, partially expressed, or some blend or mix of one allele and the other allele is an outcome. So we do not have either neither red or white flowers, but we have a blend. We have something between intermediate phenotype. In our case, pink colors. But the genotype is also the same as we have in first filial generation on left side. So heterozygous are here. Okay, so first Mendel's law describes one more time that if we cross and combine two homozygous from different pure lines, so one dominant and one recessive, we still have uniformity genotypically. So genotypes in first filial generations are same, okay? Phenotypes, so the appearance, the traits which are observable, can differ from parents, but even those phenotypes will be uniform, same between offsprings. All of the children or offsprings will be pink, will be in pink color if there is an incomplete dominance, and all of the offspring will be yellow if there is a complete dominance. Okay, so there are no differences neither in phenotype nor in genotype in first filial generation. Okay, so this is very important to know. Okay, so this is why we call it the law of uniformity. So first filial generation is uniform. Okay, only difference between is that if the trait uh, exhibits incomplete dominance, the generation will have this intermediate phenotype. Okay, so something what neither father or mother had before. Okay, so this is a pink color. We had the, let's say, mother was red, the father was white. Okay, so we had the blend if there is incomplete, because something between red and white is an outcome. If there is a complete, so dominant allele will mask the presence of recessive allele in the offspring, so that's why everything is yellow. That brings us to the second filial generation and the second Mendel's law. Guys, have you found out anything regarding second Mendel's law?
Second Mendel's law is called segregation, and segregation is that genes in the chromosome enter different cells. That's all.
Okay, yeah. The law of random segregation of genes into gametes. Okay, so let's discuss it in a little bit more detail. So, where to start? The law of random segregation of genes into gametes. When two heterozygous individuals are crossed, here we see heterozygous individuals. Those are our individuals from previous first filial generation. So we are now crossing two offspring, basically, which are heterozygous. Okay? We have two kind of alleles in the trait. Dominant one, recessive one. Dominant one, recessive one. Okay? In the previous law, or basically when we crossed two homozygous, we had in dominant homozygous we had only one type of allele. The dominant alleles were here. Okay? In recessive one, there were only recessive alleles. Now, when we are crossing two heterozygous have two possible alleles. Dominant one, recessive one. Dominant one, recessive one. And when two heterozygous individuals are crossed, each of two alleles, dominant and recessive, can be transmitted to the offspring with equal probability. Therefore, there is genotypic and consequently phenotypic segregation. What does it mean? If we combine dominant allele with dominant allele from both heterozygotes, we uh, we have one dominant homozygous. One dominant homozygous. Okay? If we combine one recessive allele with one dominant allele, we have one heterozygous. The same applies here, recessive allele with dominant heterozygous. But if we combine one recessive allele from heterozygous and one recessive allele from another heterozygous, which both are yellow, we have one homozygous recessive one, which is different phenotypically, so observable traits, phenotypically different from other offsprings and from the parents. Okay, in the other case, if we are dealing with two pinkish heterozygous flowers, and those alleles are the same as in the previous example, dominant recessive allele, and we combine two dominant, we get back the phenotype which was not present in the first filial generation. However, basically the phenotype of a grandfather or grandmother will be now present because we are now crossing two heterozygous, and when two dominant alleles are combined, the phenotype in in case of incomplete dominance is different than phenotype of heterozygous. So now we are combining dominant allele with recessive one, we have heterozygous. Recessive allele with dominant allele, heterozygous, so pinkish. But recessive with recessive, we have white flower. Because if we have a trait where the two alleles are recessive, in case of incomplete dominance, even this genotype will have a different phenotype or manifestation than heterozygous. So what does it mean, guys, basically? For us, it means that the probability phenotype, how will those flowers look like, okay, in the offspring? Probability for homozygote, dominant homozygote is 25%. Probability for recessive homozygote is also 25%. Okay? But probability to have heterozygous in the as the offspring is 50%. Okay, so our ratio or genotypic segregation ratio in our case is 1 to 1. Okay, let me use different color. One for red flower. This is genotypic segregation ratio. Okay, one for, let's use purple or pink, and one for white. In case of incomplete dominance. Okay, that was the phenotype. Guys, what is a phenotype segregation ratio in case of complete dominance? If you see the phenotypes here, what would be phenotypical ratio in case of complete dominance? Try to look here in our table here, and we see the differences in color. Okay, so we have one green, three yellowish. They are genotypically different. However, what is related to phenotypes or regarding phenotype, what would be ratio for phenotype? We had in incomplete dominance one red flower, two pinkish, and one white. How it would be the ratio in case of complete dominance?
Yellow phenotype is 3 and green phenotype is 1. So,
Perfect. Go on, Misato.
oh, I don't know how to pronounce this.
Ratio, you mean?
Yes,
Perfect. Absolutely correct. So phenotypical ratio would be 3 to 1 because in the offspring there will be probability basically that 75% for yellowish offspring and 25% for green one, or 3 to 1. Okay, perfect. Great job, guys. Perfect. Now, that was phenotype. What about genotypical ratio in complete dominance? If you again just see the table, what would be the ratio?
one ratio 1.
Okay, so we have one dominant, this one, okay? We have one dominant offspring. Then we will have two heterozygous and then one recessive homozygous. So that would be our ratio for genotype. Okay, genotype. One recessive homozygous, two heterozygous and one dominant homozygous. So this was phenotype 3 to 1, how they look, and genotype, how they are genotypically distributed, the alleles. Guys, as you see, in our case here, in the first Mendel's law, and also in our second, if there is a complete dominance, it doesn't matter if there is a dominant homozygous or heterozygous. Phenotypically, they are the same, okay? They look the same. The genotype is, however, different, but regarding phenotype, they're the same. And that is also here, okay? Even though genotypically we have ratio 1 to 2 to 1, so 1 dominant homozygous and 2 heterozygous, uh, the phenotypically they are the same. So that's why phenotype ratio is different to genotype. We have 3 phenotypes which are same and only one is different, the recessive homozygous. But in complete dominance case, if we are dealing with incomplete dominance case, we see that the phenotype is 1 red dominant homozygous, 2 heterozygous, which are pink, and 1 white homozygous. What would be genotype in this case? Genotype ratio. If we see genotypes, 1 dominant homozygous, 2 heterozygous, and 1 recessive homozygous, what would be genotypic segregation ratio?
1
1 to 2 to 1.
Perfect. Exactly. So it would be same as phenotype. Okay. What's wrong? What's wrong? So genotypically, in incomplete dominance case, genotypic segregation ratio would be the same as phenotypic. Okay, so 1 2 to 1. I will just write genotype, okay? Genotype here. So this is very important because there is a difference between complete and incomplete dominance regarding phenotypes. But as you see here, genotypically the segregation ratio is the same. So regarding genotypes, we don't care basically, we do not care whether the relation between alleles is complete dominance or incomplete dominance. Regarding genotypes, it doesn't matter. The segregation ratio is same. Phenotypically, the segregation ratio differs, and now regarding phenotypes, it very matters whether we are dealing with incomplete or complete dominance relation. Guys, so far understandable? Yes? Can we move on to the last law? Okay, so this is then move to the third law. Who was responsible for third law, guys?
Mendel's third law is one of Mendel's law of inheritance, which states that when a hybrid is created by focusing on two or more alleles, each allele is inherited independently of the others. This is true only when each allele is contained in a separate homologous chromosome, which is not the case when the alleles are linked. Therefore, it is the most exceptional of Mendel's laws. That's
Mm-hmm.
all.
Thank you very much. Now, you are absolutely right. So let me maybe provide you with a little bit more information or a little bit additional information. When we have two alleles which are examined simultaneously, the same regular segregation occurs. So if we have two polyhybrids, as we see AB, so far we were dealing just with one trait with A letters. Okay, so basically only about the color, but there are also other traits besides color. For example, in peas or in endu, uh, a surface, for example. Surface can be rounded or wrinkled. This is a trait, another trait, which is encoded via different allele or different gene, basically. Okay, so that's why we use another letter now, B. Okay, if we are checking or observing basically two traits, I hope you remember that we call it dihybrid. Okay, dihybrid because we are observing two traits. If we would be dealing with another additional trait, it would be trihybrid. In general, when we are observing more traits, we call it polyhybrid. Okay, so more than one, we already can call it polyhybrid. But uh, just to understand the differences in these little nuances in genetical terminology. As we see, we if we have two dihybrids AABB, each can form four different gametes: dominant ABs, then heterozygous AB, different heterozygotical AB, and then recessive gametes AB. So, thus, when these two gametes are crossed, they produce 16 different zygotic combinations or genotypically different, 16 genotypically different genotypes. However, some combinations are repeat so that only 9 different genotypes are eventually produced. Sorry, nine different genotypes. Let's discuss what's going on. Okay. Um, if we combine two dominant gametes, our outcome is yellowish rounded P with genotype AABB. Yeah, both all of the letters are big because both gametes were dominant. Already, however, if we just in one gamete have a recessive allele, the genotype is already different. Please zoom in if it's not obvious enough, but we see that the genotypically it's already different as we see, because comparing to the first offspring, the second has one recessive allele in as a B. Do you see? Do you see? However, the appearance is similar. So we have yellowish and rounded surface. So that would be in dominant or in complete dominance, the the outcome would be that we have yellowish yellowish and rounded peas contain 9 of 16 possible genotypical offspring, so 9 are yellowish and rounded. However, if we already combine two recessive alleles regarding the surface, so the the surface trait will be combined or crossed with two recessive alleles, the outcome is different. So you see, two recessive alleles create wrinkled surface. If two recessive alleles regarding the color are crossed, as we are able to see in this case, that we have two recessive alleles in the offspring, the color is not yellowish anymore, but more green or grayish, let's say. And that is also when two recessive alleles are combined in both traits. So one offspring from 16 possible would be in different color, in more green, and in wrinkled surface, with wrinkled surface. So this is, guys, basically Mendel's third law. Okay, please write down the phenotypic ratio 9331 regarding Mendel's third law, because this ratio is many times in the admission questions. Okay, guys, so far understandable? I would like to now play one short video regarding Mendel's genetics. Would you like to see it or no? It has 3 minutes. Okay, so we will play that video now. No. You can see the video, right? Perfect. So let's play it.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase Y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited. But it all started with Mendel and his peas.
Sorry guys, my bad, I closed the wrong window. Okay, so that's basically everything for us for today. Tomorrow we will get more into depth of genetically determined diseases in humans, and we will also have some activities regarding them. Guys, was this session helpful for you? Did you learn something, or was it too overwhelming, too confusing? You didn't get a lot. How are your feelings after the session? Okay, thank you very much. So hopefully you gained some knowledge and we discussed very important part of genetics, very important topic for admissions. So guys, that's everything from my side. I'm looking forward to see you tomorrow again. I have one question before we end regarding your group. Is it possible to postpone the starting for tomorrow of 30 minutes? Is it okay for you or no? That we will start later, not at we start at 7:10, right? Usual time, 17:10 your time. Is it possible if we start at 17:30? Because I have one appointment, I really need to be present, so I'm checking whether it is possible or okay for you to start a little bit later. Yes? Okay, so all of you went for yes. Guys, thank you very much for really understanding. I will upload also the video from the session today, and that's everything from my side. So there is no homework again. You can rest and maybe recap the things what we learned today and tomorrow we will continue where we ended. Okay? Thank you very much, guys. Have a nice rest of the day and see you soon.